Here's question 3 for the secondary BLAST. Today we're using BLAST to predict genes using homology. All right, so there is some DNA sequence here. I didn't tell you where it's from, but just like in the day that you did BLAST trying to figure out, there is some DNA sequence. From which organism is this? So we don't know where it came from. We don't know what it does. We don't even know if it does anything. Maybe it's just all non-genic, meaning doesn't encode anything. Maybe it regulates the expression of some gene, meaning when should that gene be transcribed? Is it all the time? Is it in response to some environment? So right now we don't have any of those answers. So I'm going to copy that. And because the question is, well, how many genes are encoded? Okay, actually here I told you it's human. Then I want to see, does it match any gene in the database? And there are two ways of doing that. I'll show you both of them. One of them is it searches a larger number of things, but so it searches, imagine that you have different databases and one has more items than the other. So one store has more things to search through, but the other store is more specialized for the thing you actually are looking for. So maybe you were in an online store and you can narrow down by department. But right now, we're not narrowing down anything. I'm just going to paste the sequence here, and I'm going to run a BLAST-N where I have a nucleotide sequence and I want to search a nucleotide database. So I'm now saying you have an online store and you just put on the search box and you don't narrow down by department. That's analogous to this. You're looking at the non-redundant nucleotide sequence. So what items might match my query? You run that. So while that is running, I also want to show you that I could have narrowed down my search as, okay, I'm looking for the book, not the movie of whatever my query is. So let's say that there are many different items, but which are all listed in here, but maybe my first results were not what I wanted. And I'm like, no, I only want something within this realm. So if we're looking for genes, then I know what I want is something that has been transcribed and or translated. So I can go here on the either of these, but I want to make my search more all-encompassing, so I'm going to select this one. So the reference RNA sequences, this is saying that that your database that you are going to look into is a database of RNA sequence. So you're not going to match just any DNA, you're going to match either mRNA or something called cDNA, just complementary DNA. So I'll run that. And just for fun, I'm going to open a third window, and there's my search thing, which is, I told you that if there is a gene in this sequence here somewhere in there, then that DNA sequence has been transcribed and or translated. So in my first BLAST that I ran, I said I just wanted to see if there's any sequence that matches that, and if I'm lucky, it will tell me a sequence that maybe tells me the function, if it has a function. Or maybe I have lots of results that don't say anything about transcription of that. So I narrowed down, my second window was I wanted to look at only RNA. But maybe I want to also look at protein, because if this sequence is transcribed in some frame, I don't know which frame, and then is translated, then it might match a protein. So I run what's called a BLASTX So you don't have to memorize this. I'm just going to hover over. So it says search protein databases using translate nucleotide query, meaning my query is my sequence that I enter. I'm going to enter nucleotide, and then BLAST is going to translate in 6 frames that query, and it's going to take each of those 6 theoretical proteins that could exist and search a protein database. So I put nucleotide there, and amazingly enough, it looks at protein sequences. But that's because it's going to translate that first. So I run that. So let's see what I got. Those are that are really 4 possibilities, but I'm showing you 3. One of them is search just everything, all the nucleotides in the database. And sometimes you're lucky and it even tells you what it does and from which organism. But sometimes all your hits are genomic sequence, in which case you still don't know if there are genes or not. So just a moment, there's a question. We do, you do in sequence. I would do this first one and then look at the results. And if the results are satisfying, then I don't have to look further. Now let's see, how do I know if the results are satisfying? So I look down here and I go like, oh, this says, okay, something that ends in A's is some enzyme, and it says gene. Oh, good, then there is at least one gene in that sequence that I gave you. And then you go, okay, whatever the carboxylate, that's the same gene. So this might be a longer sequence that matches that. And maybe it's an ubiquitin. So it's like, oh, is this a second gene? So are there multiple genes in that sequence? The easiest way to see how many genes is to go here on the graphic summary. So let's go there. All right, so remember the top line here is the query. So that's the sequence we entered. It had 4,500 nucleotides. And out of those 4,500 nucleotides, pretty much the whole thing, see how there's a continuous line here? So there's one sequence that matched that. And if you stop moving it around, it will tell you that's the carboxylase gene. So you're like, oh, it looks like the sequence I entered has one gene. And then you could argue, okay, but how about everything else? Okay, those are sequences that also matched that. And let's pretend I didn't have the first or the second or even third and fourth lines or fifth. So let's say this one. If I look at this result, I would say this looks like 2 genes. So one gene here and then another gene here. And the reason why I know that this whole thing is only one gene is because there's this little line connecting. This is telling me that this is some sequence that shows similarity to my query. And this is also some sequence, but this is not a continuous similarity. There is a part where they are not similar, but that little line that's connecting them is basically saying that that chunk of DNA is similar to this one chunk of DNA except for that part. So how do I know it's the same sequence? If you hover over and you stop, so it will say predicted whatever E3 ubiquitin, and then I go over here, it's going to say the same thing. So this is one result. And then here's another gene. So that one was the E3 ubiquitin. And this one is that the carboxylase gene from some other organism. So you could think maybe it's 2 genes, but because I know it's human and there is this one search that's my best match, then I'd say there is one gene in the sequence that I gave you. The other thing is that these guys say predicted. Oh, this one doesn't. Okay, so this is some other gene. So let's see. Oh, well, it says genomic sequence. So you don't actually know if it's a gene or not. Whenever it says genomic, it means that it could be a gene, it could not be a gene, we don't know. And when it says predicted, it means that we think because this is similar to that sequence, it may do the same thing, but you don't have biological data, you don't have that mRNA from the lab to know. Let me just see what the questions are. Right, so what we look for is how many lines do we have and that match here. You're going to have other, so there will be multiple exercises doing the same thing. And what we're looking for is how many lines do we have matching. If I didn't have these three and I only had that and that, then I would say there are two genes. This one is one gene, this is another gene. This one is, let's see, no, this is genomic, then I cannot say that that's a gene. When it says genomic, it's just some sequence that matches that. And I'm like, okay, I don't want genomic. I only want the stuff that matches mRNA, because then I know that whatever part of this sequence is transcribed, there will be a match. Then I'll go like, okay, then let's look at mRNA only. The reason why I said let's look at first the whole database is because this database, when you are looking at everything, you have a higher chance of finding function. So somebody might have said, oh, what the gene does. So let's look at the one that you are narrowed down by mRNA. There's a bunch of predicted, which is not really giving me biological information. This is computer generated, so I'm going to uncheck those. And I'm going to look, whatever is not predicted, then I'll go like, oh, this is a good one. That's a good one. So these don't say predicted. And this actually is going to illustrate something. So this is mRNA, non-coding RNA. So that might be part of the intron. So there is RNA that does not get translated. So it's either 5' UTR, 3' UTR, or introns. And let's see, a bunch of predicted. I don't care about those. Okay, this is some other organism. It's not human, but we could see, what is it related to. But if my question, I'm telling you, it's this DNA is from humans, then the most reliable results would be to narrow down to humans. And I don't really care that much about predicted. I only pay attention to predicted whenever I didn't really get that many hits. Then I'm like, okay, then I'll settle for something that is less trustworthy. So let me see what your questions are. Okay, I'll go back to that. So let's elaborate on this. Why is the answer 1? So right now I am on the screen where the database I searched was just RNA. So I'm asking, this DNA sequence I have, which part of this DNA sequence matches RNA? And okay, we got these results, and I got rid of the predicted ones. And I'm only going to look where the human ones match. So let's look at the graphic summary. This is my query. And the part that matches the mRNA are these parts. So the boxes are the parts that this sequence, the hit is similar to your query. The lines, they are either not similar or they actually don't even exist. It could be that on the mRNA, this nucleotide is right next to this nucleotide. So this part of the DNA sequence gets spliced out. So this guy could be an intron. So either it's something that is not similar to that, or it's an intron. And how many genes are there? So this is one gene. There's all linked by these lines that could signify introns of that gene. The boxes will be your exons because they are sequenced from your genomic sequence that ended up in the mRNA. And let's see. And when I say, okay, the answer is 1, because in this part of my genomic sequence, there's one gene encoded. It has variants, right? So if I go slowly, this one, there will be the different transcripts. But even though, so remember there are 4 transcripts, even though there are 4 variations of it, we say it's only one gene. It's that gene for that decarboxylase or whatever that was. And this last one, it's probably the other species. Let's see. Yeah, that's the Pongo. This one is not human. So I could actually not even have checked that. I was just curious to see if this other organism would have an mRNA that matches human genomic sequence. So why is it? Is the answer 1? Because within that whole query, there is one part of it that matches mRNA. And it matches different variants of mRNA. You could think about them as different alleles even, but the gene is only one gene. And just out of curiosity, let's see if the BLASTX finished running. This one, the BLASTX, we entered nucleotide sequence and we said translate it into 6 frames and tell me if it matches any protein when it's translated. And okay, unnamed, that means like we don't know what it does, but here you go, one form. So this is some allele of that enzyme. So all of these are interesting, except the unnamed. I don't care about that one. And I don't care about the predicted either. Okay, then let's look at the graphic summary. All right, this is more intense than I wanted to do right now, but this is part of also BLAST 1. Let's go down here. This part would make me think that there are 2 genes, right? One gene encoded here and one gene encoded here. But it looks like it is actually the one gene. Maybe this is the problem with this is in the sequence being compared to. Let me see what this is. This is unnamed protein add. So this is probably not a full sequence, and that's why it's giving me this break here. But the answer is one gene, because out of this whole DNA sequence, only this part becomes mRNA, different versions of the mRNA. Let me see. Can you give us a few concise steps for each program to use? Yes, let's see. Quick summary. Yeah, so what you will do is when I say how many genes are there, let me go back and maybe I'll start a new recording